Hello, my name is Christian Humphries, and my research project focuses on the use of manganese complexes in antimicrobial photodynamic therapy. To begin understanding this, it's important to first understand the importance of antibiotics to modern medicine. Since their introduction, antibiotics have been estimated to have increased average life expectancy by up to 10 years. However, this progress is currently being threatened by rapidly adapting strains of bacteria. Because of this, it's important now more than ever for us to find antibiotic alternatives with the potential to overcome this bacterial adaptability. And this brings us to photodynamic therapy, an experimental form of cancer treatment which uses light-sensitive chemicals called photosensitizers to kill cells through something known as the photodynamic effect. Now, the photodynamic effect, as you can see here, is essentially the reaction that takes place between photosensitizer and surrounding oxygen molecules when exposed to particular wavelengths of visible light. This reaction produces reactive oxygen species capable of killing cells and their DNA. Now, this has the potential to overcome bacterial adaptability through its destruction of the cells. And because photosensitizer is relatively harmless prior to irradiation, it can be concentrated in pathogenic and unhealthy cells before being irradiated, and thus avoiding healthy cell death. Now, there are many different photosensitizers currently in use, and some have been shown to be capable of killing bacteria. But for the purposes of this experiment, I will be using a manganese complex as my photosensitizer. This is because of the precedent set in nature by its use in the process of photosynthesis, as well as the fact that it is found relatively abundantly throughout the human body and is relatively harmless. Now, the goals of this project are to synthesize 2 manganese complexes for use in photodynamic therapy, to perform a structural analysis to see if they have the predicted structures, to perform an in vitro experiment to see if reactive oxygen species are formed upon exposure to particular wavelengths of light, and finally to repeat this experiment now in the presence of gram-positive and gram-negative bacteria to see if an antibacterial effect is observed. Now, once the complexes were synthesized, as you can see, they had a very different coloration and texture to the manganese chloride they were synthesized from. Furthermore, as you can see here, these are the chemical structures of the complexes that were confirmed via infrared spectroscopy, and they clearly show the form of a metal complex with the metallic atom here in the center surrounded by organic ligands. Now that I knew the structure was as predicted, I needed to know whether or not it was maintaining this structure when dissolved in solution. This is important because the experiments involving the photodynamic effect require the complexes to be dissolved. Now, as you can see from this UV visible spectroscopy, both of the complexes had different spectra from the ligand and manganese chloride they were formed from, thus implying that they are maintaining their original structure even when dissolved. Now that we knew this, I was able to perform an experiment to see if reactive oxygen species were being produced when the complexes in solution were being exposed to particular wavelengths of light. As you can see, while complex 1 showcased a steady production of reactive oxygen species over time, complex 2 did not show any significant production. For this reason, it was decided that only complex 1 would be used in the final biological tests. Now, for the biological studies, samples of gram-positive and gram-negative bacteria were exposed to complex 1. Some of these were then kept in absence of light, while others were exposed to the same frequencies of light utilized in the reactive oxygen species test from earlier. Now, we did see a significant decrease in bacterial growth in all of the samples exposed to the complex. However, there was no significant difference between the samples kept in absence of light and those exposed to light, thus showcasing that it could not have been a photodynamic effect killing the bacteria. So in conclusion, I was able to successfully synthesize complex 1 and 2. I was able to confirm their structures through structural analysis, and I was able to confirm that complex 1 was capable of producing reactive oxygen species and a photodynamic effect. However, the final biological studies showed that the bacteria were not being killed by a photodynamic effect, and thus manganese complexes would not serve as a good candidate for antimicrobial photodynamic therapy. However, this experiment could potentially be reattempted in the future using a different ligand in the structure of the complexes. This is because this would change the photoreactive properties of the chemical and therefore maybe produce a different effect in the biological studies. Now, as for my acknowledgements, I would like to thank the Appalachian College Administration for their financial assistance through the Ledford Scholarship. I would like to thank Rabo Pharmascience for allowing the use of their infrared spectroscoper in the experiments. I'd like to thank Dr. Nambiava, my project advisor, for his assistance throughout the process. And I would like to thank Brevard College for allowing the use of their chemistry laboratory for the experiments. Thank you.